In the last series of lectures, what we've done is we've looked at how essentially a sinoatrial node cardiac muscle fiber generates its own action potential. And then we've looked at how that action potential progresses and propagates throughout all of the contractile cardiac muscle fibers of the heart. And then that causes all of these, the action potential causes all of these contractile cardiac fibers to contract. So a simpler way of saying this is every single time a sinoatrial node cardiac fiber generates an action potential, and they all do at the same time, but every single time that a sinoatrial node cardiac muscle fiber generates an action potential, that action potential propagates throughout all of the cardiac muscle fibers, contractile cardiac muscle fibers of the heart, and thus they all contract. An even simpler way of saying it is every single time the sinoatrial node generates an action potential, the whole heart contracts. Now, what we need to focus on more heavily in this lecture is how an action potential traveling down a contractile cardiac muscle fiber actually translates in to that contractile cardiac muscle fiber contracting, right? This is called excitation contraction coupling. We did it with skeletal muscle fibers, right? You know how an action potential propagating down a skeletal muscle fiber causes it to contract, right? The action potential propagates down the fiber into the T-Tubule, opens DHP receptors, which pull on Ryanodine receptors, calcium spills out of the SR, and then calcium binds to troponin, myosin and actin are able to interact, etc., right? So what we need to do is do that, right? Look at how an action potential causes a contraction, but more specifically, look at how an action potential causes a cardiac muscle fiber to contract and not a skeletal muscle fiber. You're going to see a lot of similarities, but there's also some pretty big differences. So just to be clear, so we're all on the same page, what's happening here is that a sinoatrial node fiber, right, cardiac fiber, generated its own action potential. We're not going to go back through that whole thing, but it generated its own action potential. The action potential travels down the sinoatrial node fiber, and the sinoatrial node fiber is attached to a true contractile fiber right here that is part of the right atrium, right? They're both part of the right atrium, but just to be clear, this is a contractile fiber of the right atrium. All right. So they're attached to each other through an intercalated disc, and one of the things that makes up an intercalated disc is a gap junction. So if the action potential travels down the sinoatrial node fiber, then the action potential, which is just a wave of positivity, calcium ions in this case, can travel through the gap junction into the contractile fiber that the SA node fiber is attached to. And then what will happen is that action potential moving into the wave of positivity, moving into the contractile fiber will depolarize it to threshold. And then the contractile fiber will generate an action potential too. And the action potential will travel down the contractile fiber, and then it'll move into the next contractile fiber due to the gap junctions that attach the contractile fiber to the next contractile fiber, and then the action potential will move into the next and the next and the next contractile fiber. Okay, again, all that stuff you've seen already, so I'm not going to relitigate any of it. What we're focusing on here, though, is the action potential traveling down a contractile fiber. We're recognizing that this action potential is going to get passed from one contractile fiber to the next, to the next, to the next, to the next. But how does this action potential cause these different contractile fibers to actually contract? That's the part that we need to look at. So this is an image you've seen before. We're going to pretend that this fiber over here on the left is an SA node fiber, and this fiber over here on the right is a contractile fiber. Okay. And they're attached to each other through, of course, an intercalated disc, which is made up of gap junctions and desmosomes. I kind of drew that in the opposite order, so These, of course, I think you know, are the gap junctions, and these are the desmosomes. So what happens here? Well, you've seen all of the important details already in previous lectures. So again, they're attached to each other through, the SA node fiber and the contractile fiber, they're attached to each other through an intercalated disc, which is made up of desmosomes and gap junctions, more importantly, for our interests. Anyways, the action potential propagating down the SA node fiber is made up of calcium ions, right? This is the wave of positivity, the action potential propagating down the SA node fiber is made up of calcium ions. And the calcium ions are going to get pulled from the SA node fiber into the contractile fiber. Why? Because the contractile fiber has a resting membrane potential of negative 90 millivolts, and that's going to pull these calcium ions from the SA node fiber through the gap junctions into the contractile fiber. And that's going to depolarize the contractile fiber because the contractile fiber is going to start gaining all these positively charged ions. And it's going to depolarize the contractile fiber to threshold, which consists of driving the interior up by about 15 millivolts. So let's say from negative 90 up to negative 75. That was a bad 7, right? 75. Then the contractile fiber hits threshold, all things you've seen before. And at negative 75, what happens at threshold? Well, first, voltage-gated sodium channels open up. Sodium shoots in. And that drives the interior, let's say, from negative 75 up to negative 50. At negative 50, I think you know, voltage-gated calcium channels called L-type channels open up, and calcium shoots in. Now at negative 50, both calcium and sodium are going to be shooting in, working together to drive the interior up to, let's say, positive 10. In the adjacent portion of the contractile fiber, it's still negative 90, so this action potential now, this non-decremental wave of positivity, non-decremental wave of electricity, whatever you want to call it, made up of sodium and calcium ions, is going to move into the adjacently negative region and depolarize that region easily, of course, to negative 75. And then the process will repeat, right? First, voltage-gated sodium channels will open up. Sodium will shoot in. That will drive the interior from negative 75 to negative 50. Then the L-type voltage-gated calcium channels will open up. Calcium will shoot in with sodium and drive the interior up to positive 10, right? This is how the action potential propagates down the contractile fiber, okay? And then the action potential, as you know, is going to move into the next contractile fiber, and the next, and the next, and the next, okay? You've seen all of this. None of it is new. Okay? What we want to do is we want to look at how this specific sequence of the action potential propagating down this cardiac contractile fiber causes this cardiac contractile fiber to contract. Because if you can apply this and understand how this action potential causes this one cardiac fiber to contract, then you can understand how it causes all of the other contractile cardiac fibers in the heart to contract. Because ultimately, the SA node fibers are all going to simultaneously generate some action potentials. And those action potentials, one way or another, not one way or another, you know exactly how it happens, but those action potentials are going to get into and travel down all of the cardiac muscle fibers in the entire heart, right? Of the right and left atrium and of the right and left ventricle. And these action potentials are going to cause all of these cardiac muscle fibers to contract. So if you can understand how this action potential causes this one cardiac muscle fiber to contract that you're about to see, you can understand how it causes all of them to contract. This is how your textbook depicts it, and they, I would say, oversimplify it and make it wildly confusing. So we're not going to use this. I might use it just, on a real basic level, but I'm just showing you this. What this is the actual SA node fiber that it's trying to depict, and then you can see that it's attached to the contractile fiber through an intercalated disc, and more importantly, a gap junction. Okay, and it's showing you in this image exactly what I just showed you a second ago. Okay? The action potential moving from the SA node fiber here into the contractile fiber. And now the contractile fiber is generating and propagating an action potential, and it's trying to show you how that action potential causes this contractile fiber to contract, but it leaves a lot to be desired. Okay, we are going to do as I oftentimes do and just draw this out and discuss it ourselves. So to really make sense of how an action potential causes a contractile cardiac muscle fiber to contract, we need to talk a little bit about the anatomy of a cardiac muscle fiber. And you're in luck because you've seen a lot of this before. So this is an image of two cardiac muscle fibers attached to each other through an intercalated disc. Okay. What's the intercalated disc made up of? Well, desmosomes and gap junctions. Up here you can actually, this particular text, this is not from your book, but this particular text actually shows that an intercalated disc is actually made up of a ton of gap junctions, right? Not just one. And it's actually made up of a ton of desmosomes as well, not just one. Okay, so maybe that's helpful on its own to show you that an intercalated disc is actually made up of a lot of gap junctions. It's not just one gap junction, it's not just one desmosome. Anyways, the big point of showing this to you is down here is an image of another cardiac muscle fiber. And the big point to drive home is that that cardiac muscle fiber should look incredibly similar. It should look incredibly familiar to something that you've seen before. And that is a skeletal muscle fiber, right? Because if we look at the cardiac muscle fiber, what's inside of the cardiac muscle fiber? Well, it's made up of myofibrils. Cardiac muscle fibers are made up of myofibrils, just like skeletal muscle fibers are. And what are the myofibrils made up of? Repeating segments of sarcomeres. Okay? So cardiac muscle fibers are, if you look inside of a cardiac muscle fiber, the most common organelle is going to be a myofibril, right? It's going to be packed with myofibrils. Myofibrils are made up of repeating segments of sarcomeres, just like a skeletal muscle fiber. Okay? So all of that anatomy between cardiac and skeletal muscle fibers is exactly the same. They're both made up of myofibrils, and the myofibrils are made up of repeating segments of sarcomeres. And look at this. Both skeletal and cardiac muscle fibers, their plasma membranes invaginate into the muscle fiber itself. What do we call this? We call this a T-tubule. Cardiac and skeletal muscle fibers both have T-tubules. Okay. So again, this is an image. I mean, it should look incredibly similar to a skeletal muscle fiber because it is. The plasma membrane, called the sarcolemma, actually moves into the cardiac muscle fiber and runs down approximately kind of near the Z disc. Okay? And where the plasma membrane moves into the cardiac muscle fiber is called the T-tubule, just like in a skeletal muscle fiber. And what exists on the left and right side of the T-tubule within the cardiac muscle fiber? Well, the sarcoplasmic reticulum. And what's inside of the sarcoplasmic reticulum? Calcium. The sarcoplasmic reticulum lies on the left and right side of the T-tubule in cardiac muscle fibers, just like it does in skeletal muscle fibers. You're going to see it's not as dense and it's not filled with as much calcium, but overwhelmingly, the anatomy between cardiac muscle fibers and skeletal muscle fibers is identical. Okay, if you just look at like the intracellular anatomy, it's so similar. Right? There are differences between, like if you look at cardiac muscle and skeletal muscle histologically, cardiac muscle branches, and you can actually see the intercalated discs, which kind of gives it away that one is cardiac and one skeletal muscle. But if you just were to look inside of a cardiac versus a skeletal muscle fiber, they would be incredibly similar. Right? It'd be almost impossible to tell the difference, right, if somebody wasn't trained, because they're just, phenomenally similar. Cardiac and skeletal muscle fibers both are made up of myofibrils, which are made up of repeating segments of sarcomeres. They both have T-tubules. They both have sarcoplasmic reticulums, right? So that should tell you the way that an action potential is going to cause a cardiac muscle fiber to contract is going to be very similar, but some differences in the way that it causes a skeletal muscle fiber to contract. So let's go ahead and take a look at how the action potential causes the cardiac muscle fiber to contract, and we can also contrast with a skeletal muscle fiber. So the reason for going through all of that is because when I draw this contractile cardiac fiber, I draw its plasma membrane, I'm also going to draw the T-tubule here, right? That's the T-tubule, because cardiac muscle fibers, actually whether they're contractile or not, have T-tubules. The SA node fibers have T-tubules, although they're less relevant. Talk about that a little bit later. So when I draw this contractile cardiac fiber, we can draw a T-tubule within it, just like we would draw a T-tubule within a skeletal muscle fiber. Okay, now the action potential is propagating down the cardiac contractile fiber. You know exactly how that happens, right? The calcium and the sodium ions move into this negative region to polarize it. Then what happens? Well, first, what's going to happen? Voltage-gated sodium channels are going to open up. When we get to like -75, sodium is going to rush in, drive us up to -50. Then at -50, what's going to happen? These L-type voltage-gated calcium channels are going to open up. Calcium is going to shoot in with sodium. The two of them together are going to drive the interior up to like +10. Okay, now this is how the action potential is propagating down the contractile cardiac fiber with sodium and calcium. And if the T-tubule is an extension of the plasma membrane, as we've discussed many times throughout this course, then what must be located in the T-tubule? Well, in the T-tubule, we're going to find both voltage-gated sodium channels and voltage-gated calcium channels. And underneath the T-tubule right now, it's presumably still -90. So what's going to happen? This wave of positivity, which is a product of calcium and sodium ions, also called the action potential, right, is going to move into this next subsequently negative region, which exists underneath the T-tubule. And this wave of positivity, the sodium and calcium ions, as they move into this region underneath the T-tubule where it's -90, it's going to depolarize that region. And when that region becomes depolarized, let's say we get up to like -75, what's going to open first? Well, the voltage-gated sodium channels in the T-tubule are first going to open. Sodium is going to shoot in, right? Sodium is shooting into the cardiac muscle fiber, contractile cardiac muscle fiber, underneath the region of the T-tubule. That's going to depolarize it to -50. Then when we get to -50, what's going to happen? The voltage-gated calcium channels are going to open up. Calcium is going to shoot in with sodium, and the two of them together are going to depolarize the region underneath the T-tubule here to +10. And then we can assume, right, that the action potential is going to continue traveling down the T-tubule, because in the more distal region of the T-tubule, it's still going to be -90 millivolts, right? So what's going to happen? This wave of positivity due to calcium and sodium ions, this action potential, right, is going to keep propagating down the T-tubule. This region of positivity is going to move towards a subsequent area of negativity, and the same exact thing is going to happen, right? First, voltage-gated sodium channels are going to open up, then voltage-gated calcium channels, these L-type voltage-gated calcium channels are going to open up. And that's going to cause the action potential to keep propagating down the T-tubule. And this is where you might ask, in skeletal muscle fibers, as the action potential propagates down the T-tubule, what happens in the T-tubule? Well, in skeletal muscle fibers in the T-tubule, what opens up are voltage-gated calcium channels, right? Called DHP receptors. Let's draw that. Because let's make a comparison here, because this is really important to differentiate between. If this is a skeletal muscle fiber, right, in the T-tubule, right, as the action potential is propagating down the T-tubule. First of all, within skeletal muscle fibers, the action potential propagates only utilizing sodium. Okay, so only utilizing sodium, right? Sodium is going to be coming in through all these regions, driving the interior up to positive 10 or whatever, moving into the next region, underneath the T-tubule where it's negative 90, depolarizing that region, opening up the voltage-gated sodium channels, and now the action potential is propagating down the T-tubule of the skeletal muscle fiber. And then somewhere in the T-tubule, it's actually all throughout the T-tubule, but there are these DHP, dihydropyridine receptors that exist, which are really just voltage-gated calcium channels. Okay, and the DHP receptors are attached to the RyR or Ryanodine receptors on the sarcoplasmic reticulum. And when the DHP receptors open in the skeletal muscle fibers, again, these are just voltage-gated. These are voltage-gated calcium channels. So when this wave of positivity, this action potential traveling down the T-tubule moves into the next region and depolarizes it, that causes the DHP receptors, the dihydropyridine, the voltage-gated calcium channels, whatever you want to call them, but it causes them to open up. And when they open up, they pull on the Ryanodine receptors located on the SR of the skeletal muscle fiber, opening them up, and then calcium floods out, and that results in a contraction. So you might ask, are there DHP receptors in the T-tubules of cardiac muscle fibers, right? Of these contractile cardiac fibers? Because we know in the T-tubules of skeletal muscle fibers, there are DHP receptors, right? Really the purpose of an action potential traveling down a skeletal muscle fiber is so that the action potential will propagate down the T-tubules and it'll cause the DHP receptors, these voltage-gated calcium channels, to open up. And that allows calcium out ultimately of the sarcoplasmic reticulum by opening Ryanodine receptors. Does that same process play out in contractile cardiac fibers that we've been discussing? Are there DHP receptors within the T-tubule? Does opening of the DHP receptors result in the same thing? And the answer to that is much more complicated than it might seem. So let's take this one by one. In contractile cardiac fibers, are there DHP receptors in the T-tubules? And the answer to that is yes, there are. But you've already seen them. And they actually exist all throughout the plasma membrane. Okay? And how could that be? Well, every single one of these L-type voltage-gated calcium channels is actually a DHP receptor. It's a type of dihydropyridine receptor. Every single one of these L-type voltage-gated calcium channels within the contractile cardiac fibers, plasma membrane, and thus T-tubule, which is just an extension of the plasma membrane, every single one of these L-type voltage-gated calcium channels, every single one of them is a DHP receptor. So these DHP receptors, these voltage-gated calcium channels, have been opening up down the entire length of the plasma membrane and down the entire length of the T-tubule of the cardiac muscle fiber. Again, the contractile cardiac fiber. Okay. So anatomical difference number one. DHP receptors, dihydropyridine receptors, are located all throughout the plasma membrane and T-tubule of cardiac muscle fibers. In skeletal muscle fibers, we only find them in the T-tubule. What's a second huge difference? In contractile cardiac fibers, and again, I don't want to confuse you, but this applies to the SA node fibers too, because they actually propagate their action potentials only using voltage-gated calcium channels, these DHP receptors. But we're focusing on contractile cardiac fibers. So within these contractile cardiac fibers, the DHP receptors also allow calcium in to the cardiac muscle fiber. Right?
When the DHP receptors open, these L-type voltage-gated calcium channels, which are a type of DHP receptor, when they open up, they actually allow calcium in. In skeletal muscle fibers, the DHP receptors only exist in the T-tubule, and when they open up, they don't really allow calcium in. They just tug on the Ryanodine receptors, and that then allows calcium out of the SR within the skeletal muscle fiber. Let me try to emphasize what I'm actually saying here. So, as the action potential is propagating down the contractile cardiac fiber, what's happening? First, always the voltage-gated sodium channels open up. That allows sodium to shoot in, drives the interior, let's say from -75 up to -50. Okay. Then at -50, what opens up? Well, the voltage-gated calcium channels, these L-type calcium channels, which are actually DHP dihydropyridine receptors. Okay, so they open up at -50, and they actually allow calcium to shoot in. The calcium shoots in with the sodium to drive the interior up to +10. Okay, and then that keeps happening in every single region. First, the voltage-gated sodium channels open up, drive the interior to -50. Then the L-type voltage-gated calcium channels, the DHP receptors, open up. Calcium shoots in with sodium, and the two of them together drive the interior up to +10. The calcium comes in through the voltage-gated calcium channels, the DHP receptors, the L-type channels. Again, many different terms, all kind of meaning the same thing. And the sodium comes in through the voltage-gated sodium channels, and the two of them coming in together drive the interior up to +10. So do you see that within contractile cardiac fibers, not only are the DHP receptors located all throughout the plasma membrane and T-tubule, but when they open up, they actually allow calcium into the contractile cardiac fibers. That is an enormous difference between contractile cardiac muscle fibers and skeletal muscle fibers. In skeletal muscle fibers, the DHP receptors are only located in the T-tubule, and when they open up, they don't really allow calcium into the skeletal muscle fibers. They just pull on the Ryanodine receptors. An opening of the Ryanodine receptors then allows calcium out of the sarcoplasmic reticulum and allows the skeletal muscle fiber to contract. So those are the major differences with regard to where the DHP receptors are actually located in skeletal versus cardiac muscle fibers, and as well as their overall functionality in terms of either actually allowing calcium in or not. Okay, so the next thing to answer is, what about the sarcoplasmic reticulum? Okay, what's the relationship between these voltage-gated calcium channels, these DHP receptors, and the sarcoplasmic reticulum? Because we know that the sarcoplasmic reticulum exists within cardiac muscle fibers. So is there a relationship between the two? And the answer is yes. We need to address that. So let's go ahead and draw a cardiac muscle fiber. There's its plasma membrane, there's its T-tubule, and let's also recognize that sarcoplasmic reticulums exist within contractile cardiac muscle fibers. Great. All right. Next, the sarcoplasmic reticulums themselves have Ryanodine receptors located on them, or RyR receptors. This is exactly the same as skeletal muscle fibers, because if you look in a skeletal muscle fiber, you'll find that sarcoplasmic reticulums exist within skeletal muscle fibers, and located on the SR's membrane are Ryanodine receptors. So both cardiac and skeletal muscle fibers have sarcoplasmic reticulums inside, and both of them have Ryanodine receptors located on their sarcoplasmic reticulums. Okay, next, in skeletal muscle fibers, the sarcoplasmic reticulum, is physically attached to the DHP receptors within the T-tubules. That's true for skeletal muscle fibers. The Ryanodine receptors, if this was a skeletal muscle fiber, the Ryanodine receptor would be physically attached to DHP receptors located within the T-tubule, if this was a skeletal muscle fiber. That's not the case for cardiac muscle fibers. Okay? In cardiac muscle fibers, the Ryanodine receptors are not physically attached to the DHP receptors whatsoever. Okay? Where are the DHP receptors? The DHP receptors are located all throughout the plasma membrane and all throughout the T-tubule. These are all DHP, a type of dihydropyridine receptor. We also call them L-type voltage-gated calcium channels. So DHP receptor, L-type voltage-gated calcium channel doesn't make a difference. They're in cardiac muscle fibers, contractile cardiac fibers. The DHP receptors, these L-type voltage-gated calcium channels, exist all throughout the plasma membrane and all throughout the T-tubule. Okay? And in no location are these DHP receptors, these voltage-gated calcium channels, attached to the Ryanodine receptors located on the sarcoplasmic reticulum. Okay, huge difference between skeletal muscle fibers and cardiac muscle fibers. The DHP receptors and the Ryanodine receptors are not physically attached to each other. But is there a relationship? Right, there's the next question. Is there a relationship between the DHP receptors located in the plasma membrane and the T-tubule of the contractile cardiac fiber and the Ryanodine receptors located on the sarcoplasmic reticulum? And the answer is yes, there is. There's a huge relationship. The Ryanodine receptors located on the sarcoplasmic reticulum within the contractile cardiac fiber would not open if it weren't for these DHP voltage-gated calcium channels throughout the plasma membrane and throughout the T-tubule. But there's a more important question, which is why? All right, so pretend that the action potential is propagating down the contractile cardiac fiber, right? What's going to open up first? The voltage-gated sodium channels. When you get to like -75, the voltage-gated sodium channels open up, sodium shoots in, drives the interior from -75 up to like -50, and then what happens? Well, the voltage-gated calcium channels, called L-type or DHP, dihydropyridine receptors, open up, and that allows for calcium to shoot in, and then the sodium and the calcium are both coming in and drive the interior up to +10. So that's how the action potential actually propagates down the contractile cardiac fiber, utilizing both sodium and calcium. And here we can actually answer why that's necessary. Why is it necessary for both sodium and calcium to drive the propagation of the action potential within contractile cardiac fibers? And the answer is located over here on the sarcoplasmic reticulum are these Ryanodine receptors, right, within the cardiac contractile fibers, just like in the skeletal muscle fibers. Okay, but what's going to happen is that calcium that came in, right, that calcium that's coming in, that's allowing for the action potential to propagate, right, both sodium and calcium, that calcium that's coming in, what it does is it actually goes over to these Ryanodine receptors located on the SR, and it binds to them. And when it binds to them, it causes them to open. And that allows calcium to spill out of the sarcoplasmic reticulum into the cytosol. This is a process called calcium-induced calcium release, CICR. Let's think about what it means. Calcium release. What are we talking about with regard to calcium release? Well, calcium is being released from the sarcoplasmic reticulum because the Ryanodine receptors are opening, right? This is calcium release from the sarcoplasmic reticulum within the contractile cardiac fibers. Okay, so calcium released from the sarcoplasmic reticulum. Now, next thing. Calcium-induced calcium release. Where's the calcium-induced coming from? Because the calcium that's coming in due to the action potential propagating, right? The action potential propagates with both sodium and calcium, right? The calcium that's coming in is actually inducing the calcium release. How? Because the calcium that's coming in, driving the propagation of the action potential, the calcium that's coming in is inducing the release of calcium by binding to the Ryanodine receptors on the sarcoplasmic reticulum and opening them. Okay? That's what calcium-induced calcium release means. This happens in cardiac muscle fibers. More specific, this happens in, to a degree in the SA node fibers too, but it's so insignificant compared to the contractile fibers. Okay, calcium-induced calcium release. The calcium that's coming in from the interstitial fluid into the contractile cardiac fiber, which is actually part of what's allowing the action potential to propagate. That calcium that comes in actually binds to the Ryanodine receptors located on the sarcoplasmic reticulum and opens them, and that allows the calcium out. So remember what I said. If it weren't for the DHP receptors, right, these voltage-gated calcium channels, the Ryanodine receptors wouldn't open up. That's true in both skeletal and cardiac muscle fibers, but for very different reasons. Right? In skeletal muscle fibers, the opening of the DHP receptors specifically and only within the T-tubule results in the Ryanodine receptor opening up on the SR because it physically pulls on it. Right? But within cardiac muscle fibers, that's not what happens at all. The DHP receptors are still responsible for the Ryanodine receptors opening, but for an entirely different reason. The DHP receptors, first of all, are located all throughout the plasma membrane and all throughout the T-tubule, and they actually allow calcium into the contractile cardiac fiber. And that calcium that comes in through them then binds to the Ryanodine receptors and opens them up. So it's the calcium that's coming in through the DHP receptors, the voltage-gated calcium channels, in contractile cardiac fibers that actually allows for the opening of the Ryanodine receptors located on the sarcoplasmic reticulum, and then calcium spills out. It's not that the DHP receptors are in any way, at least in cardiac muscle fibers, it's not that the DHP receptors are in any way physically attached to the Ryanodine receptors. It's the influx of calcium through them that allows for those Ryanodine receptors to open up. And now hopefully an image like this out of your textbook makes a little bit more sense. It's so oversimplified that I really I can't even use it. But I think we can make sense of it now. Okay, so what it's trying to show is, first of all, this is a contractile fiber. The action potential is propagating down the contractile fiber. How does it propagate? Utilizing both sodium. Right? Sodium comes into the voltage-gated sodium channels, driving the interior up to like -50, which causes then in this case, let's just draw them in green because that's how it's doing it. Once you get to -50, then the DHP receptors, the voltage-gated calcium channels open up. Okay? And then calcium shoots in with sodium, drives the interior up to like +10. That's how the action potential propagates. And then this calcium that's coming in through these DHP receptors, these voltage-gated calcium channels, the calcium that comes in actually travels over and binds to the Ryanodine receptors located on the sarcoplasmic reticulum, and it causes them to open. This is called calcium-induced calcium release, because when the Ryanodine receptors open up, induced by calcium, that causes calcium within the SR to be released. And then what happens? Okay. You've seen all of this before, because skeletal and cardiac muscle fibers anatomically are, again, inside almost identical. What is within a cardiac contractile fiber? Inside of the cardiac contractile fiber, you're going to find myofibrils, and the myofibrils are made up of repeating segments of sarcomeres. Within those sarcomeres, there are actin and myosin filaments, and the actin filaments within cardiac muscle fibers look just like the actin filaments within skeletal muscle fibers. They're made up of 2 F-actin strands, right, with which are ultimately all made up of G-actin, which all have myosin binding sites located on them. Then there is tropomyosin that covers up all the myosin binding sites on the different G-actins, and there's also troponin, and as long as tropomyosin is covering up those myosin binding sites on all the different G-actins, then myosin and actin can't interact. So how do we resolve that problem, right? This is exactly the same in skeletal and cardiac muscle fibers. What you need is for calcium to bind to troponin. That causes then a conformational shift in troponin, which pushes tropomyosin off of the myosin binding sites on all the different G-actins, and that enables the myosin heads to be able to interact with actin, and we call this a contraction. Okay? The release of calcium, and I have to be careful here because there's actually more to say about this, but the influx, let's just say it this way, the influx of calcium into the contractile cardiac fiber through both the calcium that's coming in through the DHP receptors or voltage-gated calcium channels and the calcium that's being released from the SR, that calcium is going to bind to troponin, cause a conformational shift in troponin, push tropomyosin off of the myosin binding sites on actin, and that is going to enable for a contraction to occur because it's going to enable for myosin and actin to interact, and that's where force is generated. Okay. This is how an action potential enables for a cardiac contractile fiber to contract, right? This is how the action potential accomplishes it. This is what we call excitation contraction coupling, but now this is excitation contraction coupling of a cardiac muscle fiber compared to a skeletal muscle fiber. There are some differences. Yes, the action potential is going to allow for a skeletal muscle fiber to contract. And the action potential is also going to allow for a cardiac muscle fiber to contract. But there are some, as you've seen now, some differences in what enables that to happen. The end result is exactly the same. The action potential in skeletal muscle fibers and in cardiac muscle fibers, the action potential in both cases is going to cause calcium levels to rise inside. And then the rise in calcium is going to bind to troponin and ultimately allow myosin and actin to interact, which is going to result in a contraction. But how you get to the point of calcium rising inside of the fiber is different in skeletal muscle fibers versus cardiac muscle fibers. So we need to talk about that just a little bit more. So in both skeletal and cardiac muscle fibers, the action potential enables for intracellular calcium to rise, and that translates into a contraction for the exact same reason between cardiac and skeletal muscle fibers. But there's a big difference in where the calcium comes from. Okay? In skeletal muscle fibers, again, I won't go over this too much because hopefully you know this from so many lectures that we did, but in skeletal muscle fibers, the calcium that rises inside is all coming from the SR, right? When a skeletal muscle fiber contracts, the action potential propagating down the plasma membrane into the T-tubule causes the DHP receptors to open, opens the Ryanodine receptors, and then calcium spills out of the SR. And that calcium that spills out of the SR is the sole reason why calcium levels increase inside of the skeletal muscle fiber. That's the only explanation. The calcium comes from the SR, and that is it within skeletal muscle fibers. Now, where does the calcium come from in cardiac muscle fibers? There's a big difference here. The calcium within cardiac muscle fibers is coming in all throughout the plasma membrane and all throughout the T-tubule. And then that calcium is causing more calcium to be released from the SR into the cardiac fiber. But there's a big question in cardiac physiology, which is, which calcium is more important? Is it the calcium that's coming in from the interstitial fluid, right, the extracellular fluid, or is it the calcium that's coming out of the SR? And here's what we know about cardiac muscle fibers. Their SRs are very weak. They're not very dense, and they don't actually store a ton of calcium. Yes,
During excitation contraction coupling, thanks to calcium-induced calcium release, calcium does get released from the SR of cardiac muscle fibers. And that calcium that's released from the SR does participate in enabling the contraction. But it seems fairly clear that the calcium that's coming from the extracellular fluid is just as, or perhaps even more important to the contraction. Okay? Meaning the calcium that's coming in as the action potential is propagating through the DHP receptors or voltage-gated calcium channels, whatever you want to call them. That calcium that's coming in, let's draw it like this, that calcium that's coming in as the action potential is propagating, that calcium that comes in serves two purposes. One, it causes calcium-induced calcium release. It binds to the Ryanodine receptors, opens them up, allows calcium out of the SR. But secondly, and probably even more importantly, it directly participates in enabling the contractions. The calcium that comes in actually, physically, this calcium is actually involved in binding to troponin and enabling for the interaction of myosin and actin. The experiments on this are pretty difficult to do, but it appears like in some of, studies don't completely agree with each other, but basically what you can try to do is you can try to knock out the entire SR, the sarcoplasmic reticulum, of a cardiac muscle fiber. Like you can try to remove its Ryanodine receptors or try to influence their opening. And the studies seem fairly clear that if cardiac muscle fibers just didn't have functional sarcoplasmic reticulums at all, they would still be able to contract and produce a good amount of force. Why? Because so much calcium is coming in from the action potential propagation, right? There's a ton of calcium that's coming in through these voltage-gated calcium channels all throughout the plasma membrane and the T-Tubule. And that calcium that comes in, a big part of it actually participates in binding to troponin, pushing tropomyosin off of the myosin binding sites on actin and allowing for a contraction to happen. And it would appear that the sarcoplasmic reticulum isn't that important. It's definitely hugely beneficial for cardiac muscle fibers to have sarcoplasmic reticulums and store calcium. But what I'm saying is some contraction for sure would still happen even if you completely got rid of the sarcoplasmic reticulums. And of course, I mean in cardiac contractile fibers, right? Let's just say you completely removed the SR, right? Just the action potential propagating down the contractile cardiac fiber, down the plasma membrane into the T-tubule. That propagation of the action potential involves a whole bunch of calcium coming in, and that calcium alone would be enough to allow for a pretty decent contraction to happen in the cardiac contractile fiber. Now, with the SR intact and thus calcium-induced calcium release happening, the contraction is much more forceful, right? Because it's obvious that this calcium that gets released from the SR through calcium-induced calcium release also is important in participating in the contraction as well. The point is, if you want like a maximal, you're going to see this isn't actually a maximal contraction that's happening right now, but I mean, like, if you want a very good contraction, a necessary contraction, let's say for survival, you really need both of them. Okay? You need both calcium that's coming from the extracellular fluid, interstitial fluid, and you need the calcium that's being released from the sarcoplasmic reticulum. The two of them combined together are what enable for a really complete whole contraction to happen. But the overarching point of this is, right, this is completely different than skeletal muscle fibers, right? Because the only place that calcium comes from in skeletal muscle fibers is by being released from the sarcoplasmic reticulum. That's it. It's the sole explanation. If you were to remove the sarcoplasmic reticulum of skeletal muscle fibers, they wouldn't contract at all. They would just be done for. But in cardiac muscle fibers, the calcium is coming from both the extracellular fluid and the sarcoplasmic reticulum. So again, the big point of all of this is to say with contractile cardiac fibers, the calcium that's coming out of the SR and the calcium that's coming from the interstitial or extracellular fluid is both, they're both very important to the actual contraction of the cardiac fiber itself. Part of the reason in figuring, scientifically in trying to figure out which calcium is more important, the calcium from the SR or the calcium from the extracellular fluid, part of the reason that it's so challenging is because, well, if you knock out, let's say, the DHP receptors, which a lot of medications can do, again, conversation for a different time, but if you knock out these DHP receptors and you don't allow calcium to come in through the DHP receptors all throughout the plasma membrane and T-Tubule, then you can't get calcium-induced calcium release, and calcium won't be released from the SR. So how can you make a fair comparison between the two? It can't be done. You need the DHP receptors to open throughout the plasma membrane and the T-tubule in order for the Ryanodine receptors to open. So if you wanted to do an experiment and you see like, okay, if we just, didn't have the DHP receptors open up anymore, or to a lesser degree, what would the contraction be like? But it's like, well, if you shut the DHP receptors, then you're not allowing calcium in. Calcium's not going to cause calcium-induced calcium release, so the calcium won't come out of the SR, and of course you're going to get a less forceful contraction. So it's a very hard experiment to do. Point being, we know that the calcium that comes from the extracellular fluid and the calcium that comes out of the SR is both very important to enabling the cardiac fibers to contract. But we can also say with regard to cardiac muscle fibers, if there was no extracellular calcium, like if you didn't exist, calcium is an electrolyte, so like let's say that there was no extracellular calcium, then there would be no calcium that could come in through the DHP receptors, the voltage-gated calcium channels. There would be no calcium-induced calcium release, and there would be no contraction, period. Right? Without extracellular calcium, cardiac muscle fibers are not contracting, period. It can't happen. Okay? You need the calcium to come in for one, to help participate in the contraction, and two, cause calcium-induced calcium release as well. So if you don't have extracellular calcium, you don't have a contraction. And you can go all the way back to the SA node and say, well, if the SA node fibers, like if there was no extracellular calcium, or levels were very, very low, how would the SA node fibers propagate their action potentials to begin with? They wouldn't. There would be no way to do it. So the SA node fibers wouldn't even be able to generate action potentials without extracellular calcium. And then those action potentials wouldn't get into the contractile fibers, period. You wouldn't have a heart rate, right? That's why maintaining the, calcium levels sufficiently within your bloodstream and thus extracellular fluid is so incredibly important. Your body will stop at no means to ensure that calcium levels stay sufficient in your bloodstream. There's calcium within every sports beverage that you drink, right? There's a reason, right? Your heart doesn't work without extracellular calcium, period. Doesn't happen. So in that regard, which calcium is more important, extracellular calcium or calcium from the SR? Obviously, it's extracellular calcium, but that wasn't actually the true part of the conversation. The true part of the conversation was there is calcium that's stored in the SR, and if you were just somehow allowed to release that calcium from the SR without any calcium coming in from the extracellular fluid, what would the contraction be like? And the point I was trying to make is, well, that experiment is unbelievably difficult to be able to do for this exact reason. You kind of need to allow the calcium in from the extracellular fluid to be able to enable sufficient amounts of calcium to be released from the SR. So it's just like an almost impossible experiment to be able to do, at least perfectly. There are ways to try to do it, but it's very hard. Last point to make here before we move on. After these fibers contract, we need these fibers to relax, right? The fibers contract and then they relax. Like if it, you can have left ventricular systole. That's when you're talking about the contractile cardiac fibers of the left ventricle contracting, and then there's left ventricular diastole when the left ventricular cardiac muscle fibers, contractile fibers, relax. Okay, so After a contraction, there has to be a relaxation. How do the fibers relax? And the answer is, well, located on the SRs, there are these circupumps, sarcoendoplasmic reticulum calcium ATPase pumps, and they're activated actually by calcium levels rising inside of the contractile fiber. And when calcium rises, it'll bind to them, activate them. At least it'll increase their activity. It'll make them more active. That's not something I'd ask on the exam, but that'll increase their activity. And what will they do? They'll kick the calcium from the cytosol, the intracellular fluid, back into the sarcoplasmic reticulum, right? They're a type of primary active transport. They're taking calcium from a lower concentration inside of the cytosol to a higher concentration in the sarcoplasmic reticulum itself, right? Because calcium levels would still be higher in the SR comparatively, we would say, to the cytosol. Okay. All right. So the circupumps take the calcium and they kick it back into the SR. That's going to cause calcium levels within the cytosol to fall. But where did a lot of this calcium come from that's inside of the contractile fiber right now? A lot of the calcium came from the interstitial fluid, right? Part of the extracellular fluid. Right? It came in, from the actual interstitial fluid itself as the action potential was propagating down the cardiac fiber. A lot of the calcium that came in actually came in from the interstitial fluid. So the point is, we need to kick that calcium also back out into the interstitial fluid. We don't just need to return the calcium to the SR like we do in skeletal muscle fibers, right? That's the only place that calcium comes from. So in a skeletal muscle fiber, you just take all the calcium and you kick it back into the SR. But in a cardiac muscle fiber like this, a lot of the calcium came from the extracellular fluid, right? The interstitial fluid. So you need to take the calcium that's in the cardiac fiber that's allowing for the contraction to happen and kick it back into two places. You need to kick it back into the SR, which is done by the circupumps, and you also need to kick it back out into the interstitial fluid. And which pumps do that? There's a whole huge cardiovascular physiology scientific debate around this. I'm not going to ask any questions about this. There's a combination of different pumps. Some of them, I'm not even going to get into it. Some of them, probably different types of active transport pumps work to take the calcium from the cytosol, intracellular fluid, and kick it back out into the interstitial fluid or extracellular fluid. Okay, that's the only point I want to make. The calcium gets kicked back out of the contractile cardiac fiber, back out into the extracellular fluid. You need to remove it two ways, right? Back into the SR and also back out into the extracellular fluid, because a lot of it came from the extracellular fluid. And then once you do that, calcium levels will fall sufficiently inside of the contractile cardiac fiber, and thus you won't have calcium binding to troponin anymore, and tropomyosin will return to its original orientation, covering up the myosin binding sites on all the different G actins. Myosin and actin will no longer be able to interact, and you won't have a contraction anymore. We call that a relaxation. So by removing the calcium back into the SR and back out into the extracellular fluid, the cardiac fiber will relax. And I realize I did not go through the repolarization process. Like, how does this cardiac muscle fiber repolarize? The repolarization process itself is quite complex. I mean, there's not like everything that we've talked about in this course gives you more than enough information. Like if I were to go through it, it would make complete sense to you, I think. For those of you that this material has really been making a lot of sense, like the repolarization process would not be a problem for you to understand. It would look very similar in a lot of ways to so many different things we've already discussed. But it takes another hour to be able to do. And then you can apply it to so many different medications that are used in cardiovascular physiology. It's an enormous amount of fun to actually discuss. It's incredibly interesting, but it's one of those things where for the sake of time, we just can't do it. There are, in fact, countless things I'm bypassing. You know, if you think of like becoming a cardiologist or something, it takes in, you have to go well beyond medical school to be able to become a cardiologist. And that's because the physiology of the heart is unbelievably complex. And you can go on and on about it for literally years and years if you wanted to. And there's so much we still don't know about it. So with the time that we have, obviously my objective is always to try to give you some foundational understanding of the material. But if this stuff interests you, I would just highly recommend that you try to take more advanced, physiology courses, maybe cardiovascular physiology courses if you have the chance. And after that, hopefully you can begin to see the whole thing. Because what happens in a normal functioning heart? The SA node fibers, right, these autorhythmic pacemaker cells, they generate their own action potentials. And then what happens? The action potentials move from the SA node fibers, right, into the contractile fibers, because the SA node fibers are attached to the contractile fibers through intercalated discs. And then the action potential moves from one contractile fiber to the next, 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 because they're all attached to each other through intercalated discs. So the action potential ultimately will move from the SA node fibers into all of the different contractile fibers of the heart. Right? I think we've gone over this in the past. But what's that actually going to result in? If the action potential moves from the SA node fibers and it propagates into all of the contractile fibers of the heart, what's the action potential going to do to those fibers? And the answer is, well, we just went through it in an enormous amount of detail. It's going to cause them all to contract through the process of excitation contraction coupling. This action potential that starts with the SA node fibers propagates throughout all of the contractile cardiac fibers of the heart and results in all of them contracting, because that's what action potentials do to cardiac muscle fibers and skeletal muscle fibers for that matter. Albeit the mechanisms are slightly different in the way that it accomplishes the contraction. But this is why these sinoatrial node fibers generating their own action potentials translates into a whole heart contraction. Every single time the SA node, these fibers generate their own action potential, that action potential propagates throughout all of the contractile fibers of the heart, and they all contract. And that's where a heartbeat comes from. Now we have to address something else, which is, why does the right and left atrium, the muscle cardiac fibers of the right and left atrium, contract before the fibers of the left and right ventricles? And to be able to discuss that, you have to begin the conversation about the dreaded conduction system. The conduction system is something you learn about in anatomy, and oftentimes it's usually you just memorize it. And because in anatomy you can't, there's no real conversation of physiology, at least to any meaningful degree. So really understanding the conduction system is just impossible to do in anatomy. You need physiology to do it. This is an image of the conduction system. Okay, I'm going to explain it as I go. What's going to happen is the SA node fibers are going to generate their own action potential. Okay? That action potential is going to begin propagating throughout all of the contractile cardiac fibers of the right and left atrium. And it's going to propagate through them so fast, right? The way that this works is the action potential actually rips throughout all of the contractile fibers of the right and left atrium so fast that they all pretty much contract in unison. Now, there are slight differences in like milliseconds when one starts contracting to the next, but, to the human eye, really the entire right and left atrium, all the fibers really contract at the exact same time. And that's because that action potential it's really fast electrical impulse. It rips throughout all the fibers so fast that they all contract pretty much in unison. But here's the thing. The left and right atrium are only attached to the left and right ventricle in one location, and that's it. You would think, right, really easily, the action potential would just begin to move from the left and right atrial fibers down into the left and right ventricular fibers. And you think, well, yeah, that's what should happen, but that's not how it works, okay? The left and right atrium is separated from the left and right ventricle by a thick layer of connective tissue. They're not actually attached to each other except for in one location. There's only one location where atrial fibers are attached to ventricular fibers, and it's at what's called the AV node. The AV node is really the one location where atrial and ventricular fibers are attached to each other. So if you want this action potential, it's a collection of them, but if you want this action potential that started in the SA node to move from these atrial fibers into these ventricular fibers, the only place that it can do that is by going through the AV node. So first of all, what is the AV node? The AV node is a collection of cardiac muscle fibers, just like the SA node is. Okay? But the AV node is also just like the SA node in the fact that these are fibers that are autorhythmic. They're pacemaker cells. They can generate their own action potentials. But the difference is the AV node fibers generate their action potentials. Let's say this, they reach threshold much, much more slowly than SA node fibers do. And we'll get to that in a moment, but I'm getting a little ahead of myself. So let's look at the connection between atrial and ventricular fibers at the level of the AV node first. So here we're going to say is an atrial fiber. And here we're going to say is an AV node fiber. Okay? This is where the atrium and the ventricles meet. Okay? It's at the AV node. Okay? The AV node is where atrial and ventricular fibers ultimately meet one another. All right, so this is an atrial fiber connected to an AV node fiber. How are they connected to each other? They're connected through intercalated discs. That means that there are clearly gap junctions between the two of them. So what's going to be happening here, right? The action potential is going to be propagating down the atrial fiber. How does it propagate? Using a combination of both sodium and calcium, right? You've seen all of this, right? And then what's going to happen? The action potential, this wave of positivity, is going to get pulled over into the AV node fiber. Now, for the moment, all we're going to say about the AV node fiber is its physiology is incredibly similar to SA node fibers. The AV node fiber has funny channels. It slowly gets to threshold on its own. It just does it a lot more slowly than the SA node fiber does. We'll talk about the relevance of that in a second. And it propagates its action potentials utilizing only calcium. Okay, now, what's going to happen here is the sodium and the calcium, the action potential, wave of positivity, is going to move into the AV node fiber. And that's going to get this AV node fiber to threshold. In this case, let's say that that's, right around like -50 or something. Okay, it gets it to threshold. And then what does the AV node fiber do? It propagates its action potential, but it propagates its action potential just like the SA node fiber does. It propagates its action potential utilizing calcium. These L-type voltage-gated calcium channels, which you now know are DHP receptors, dihydropyridine receptors. So calcium flowing in to the AV node is what enables the action potential to propagate. And then you have to ask yourself, what exists beyond the AV node fibers? What are the AV node fibers attached to? The AV node fibers are attached to, technically, these things called conduction fibers that you're about to see. But let's just say that the AV node fibers, after you get beyond the AV node fibers, you're kind of into the true contractile fibers of the ventricles. It's going to be what's called the interventricular septum, first of all. Okay, we're just going to say that these are, we'll just say it this way, they're ventricular fibers. Okay, and the AV node fibers are attached to ventricular fibers through intercalated disc and thus gap junctions. So what's going to happen? Calcium is going to move from the AV node fibers through the gap junctions into the ventricular fibers, which have resting membrane potentials, by the way, of -90, and it's going to cause them to get to threshold. And then the ventricular fibers are going to start propagating their action potentials, and they're just like atrial fibers in the fact that they propagate their action potentials utilizing both sodium and calcium. So the point is, once you get past the AV node fibers, you start getting into what, let's say, the true ventricular fibers. And then the action potential starts propagating throughout all these true ventricular fibers, and the right and left ventricle contract. But that doesn't answer the question of why there is a delay between when the right and left atrium contract and when the right and left ventricle contracts. Because we know that there's a delay, right? The right and left atrium contracts, then there's a very short delay, and then the right and left ventricle contract. In an ECG, the right and left atrium contracting is the P wave, and then the right and left ventricle contracting is the QRS complex. And we know that there's a delay between those two. So what accounts for the delay? So the SA node fibers, they generate their own action potential. The action potential from the SA node fibers then propagates, as you know, throughout all of the atrial fibers. That action potential propagating throughout all these atrial contractile fibers results in them all contracting. And they all contract before the action potential propagates throughout the ventricular fibers and causes all of the ventricular fibers to contract. Now, how is that the case? Okay, well, we know that the only place that the atrial and ventricular fibers are attached to each other is at the level of the AV node. So the action potential is traveling throughout the atrial fibers, and then the action potential enters into the AV node fibers, causes the AV node fibers to get to threshold. Then the AV node fibers generate an action potential, and the action potential travels down the AV node fibers into, let's say, what are the true ventricular fibers. And then the action potential begins propagating throughout the true ventricular fibers, and that enables these true ventricular contractile fibers to contract. But yet again, where's the delay? Most people believe that the delay happens because of the fact that the AV node fibers propagate their action potentials utilizing only calcium. Okay? If this is an AV node fiber, how does the AV node fiber propagate its action potential? It propagates its action potential utilizing only calcium. And this is what we think is generally true. When calcium is used as a means to propagate an action potential, the action potential propagates very slowly. You know, there's probably more intricacies to this, some of which we don't fully understand, but the action potential appears to propagate with calcium at a much slower rate. Okay? So the point is, when the action potential begins to propagate down these AV node fibers, it slows down significantly, right? The action potential is propagating through these atrial fibers very rapidly because the atrial fibers propagate their action potentials utilizing, most importantly, sodium and calcium. And that allows for the atrial fibers to propagate action potentials very quickly. But then the action potential, the only place, again, where the atrial and ventricular fibers meet is at the AV node. And when the action potential begins to propagate down the AV node fibers, it then just slows down dramatically because the action potential, as it propagates down the AV node fibers, begins to propagate very, very slowly. And this is what we think accounts for the delay, right? Because as that action potential is propagating down these AV node fibers, the rate and speed, the velocity at which the action potential is traveling down the AV node fibers is significantly reduced. There's it slows down the propagation of the action potential substantially. And then finally, right, it then moves from the AV node fibers through the gap junctions into, let's say, right here, the beginning of the, let's say, true kind of ventricular fibers. And then once the action potential gets into these true ventricular fibers, the ventricular fibers propagate their action potentials utilizing both sodium and calcium, and the action potential again picks up the speed and begins to propagate incredibly quickly again. And then the action potential rips throughout all of the left and right ventricular fibers, and they all pretty much contract in unison. And that's where in the ECG we see the QRS complex. Okay? So this is what we think. This is the most educated guess that we have on this right now, is that this is what we think accounts for why the right and left atrium contract before the right and left ventricle do. We think that, well, we know for one, that the atrial fibers and ventricular fibers are only attached to one another at the AV node. And when the action potential travels from the atrial fibers into the AV node fibers, the speed at which the action potential propagates is significantly reduced. It just slows down in time. And that enables for the right and left ventricular fibers to contract and kind of finish contracting, like as the action potential slowly propagating down the AV node fibers. And then, kind of we'll say that the right and left atrial fibers are just kind of finishing up their contraction, beginning, to kind of relax. Then the action potential finally gets through the AV node fibers into the ventricular fibers, and then the action potential starts ripping through the ventricular fibers again because they propagate their action potentials utilizing both sodium and calcium, and then it rips throughout all of the right and left ventricular fibers, and they all contract in unison, and that's where we see the QRS complex. So that's the first part of this, is showing you how the AV node we think accounts for the delay between atrial and ventricular contractions. So the first part of this was recognizing that the AV node fibers play a very important role in causing the delay between when the left and right atrium contract and when the left and right ventricle contract. But the AV node has more important functions than just that. The AV node does more than just cause a delay between when the atria and when the ventricles contract. The AV node is actually similar to the SA node. It's structurally and functionally, it's almost identical. The AV node is capable of generating its own action potentials, just like the SA node does. The SA node fibers and AV node fibers are both capable of generating their own action potentials. We need to talk about the relevance of that now. What's the reason that these AV node fibers are capable of generating their own action potentials? Why do we want them to be able to generate their own action potentials? And another question is, generally, why don't they ever actually get to the point of generating their own action potentials if they have the ability to? More way of saying that is they don't ever accomplish it in a healthy heart. Okay? Because in some people, they do. All right, so here is the AV node fiber. Okay? The AV node fiber is just like the SA node fiber in the sense that it has funny channels and that it can get to threshold on its own. Okay? But here's what we also think about AV node fibers. They get to threshold a lot slower than SA node fibers do. AV node fibers get to threshold a lot slower. You can spend all day theorizing about why that might be the case, and there's a whole field of study that looks into why this is, and no one's 100% certain. Many possible explanations. Okay, maybe there's I'm pausing because there's a I don't even know what to say here because there's so much variance in what's discussed, but there could be differences in the funny channels, the membrane potential could be more negative. I mean, there's all kinds of things that could cause the AV node fiber just purely, we know that it takes longer to get to threshold. Okay, let's just leave it at that. Okay, if you want to go through all the different possible theories, again, take more advanced physiology theoretical courses. Okay, so it takes longer for the AV node fiber to get to threshold. Now we need to do this actually just a little bit different. Up here is the SA node fiber. Okay. Over here is the AV node fiber. Okay. And then there's a collection of atrial fibers that exist in between the SA node fiber and the AV node fiber. All right. So let's say that the SA node and the AV node fiber are both starting to get to threshold on their own. Okay, at the exact same time. It both starts at the exact same time. But the SA node fiber gets to threshold way before the AV node fiber does. So if the SA node fiber gets to threshold, what's going to happen? The action potential is going to start propagating down the SA node fiber into the atrial fibers. It's going to be propagating throughout all of the atrial fibers. And as the action potential from the SA node fiber is propagating throughout all of the contractile fibers, the atrial contractile fibers here, here, and here, the AV node fiber still hasn't gotten to threshold. So the SA node fiber generated its own action potential. The action potential started propagating down the SA node fiber into the atrial contractile fibers. And now it's propagating throughout all of the atrial contractile fibers while the AV node fiber is still trying to get to threshold on its own. It still hasn't gotten to threshold on its own. And what's going to happen is the action potential, its wave of positivity, is actually going to head in from the atrial fiber into the AV node fiber before the AV node fiber ever gets to threshold on its own and cause the AV node fiber to get to threshold. The influx of calcium and sodium from this contractile atrial fiber into the AV node fiber is what causes the AV node fiber to get to threshold. It never actually accomplishes it by itself in a healthy heart, because the SA node fiber gets to threshold so much faster than the AV node fiber does that the action potential propagates throughout all of the atrial fibers and, contractile atrial fibers, and then into the AV node fibers, causing the AV node fibers to get to threshold before it ever does it on its own. So what's the point of the AV node fibers generating their own action potentials at all and being like SA node fibers? And the answer is, what if the SA node fiber stopped working? Because this happens to people. What if their SA node fibers just stopped generating their own action potentials? This can happen. Some cardiovascular disease causes the SA node fibers to just cease from generating their own action potentials. Okay? That would be certain death, right? Because if the SA node fibers don't generate their own action potentials, then the action potentials won't propagate throughout the heart, and the heart won't contract, and blood won't be pumped around your body, and you'll die. But what do we have here? The AV node actually generates its own action potentials. It just generates them. It takes them a lot longer to get to threshold. So if the SA node, let's say that it stopped working, the action potential wouldn't propagate throughout the atrial fibers, which isn't a good thing. So the atrial fibers would never contract. But what would eventually happen? Eventually, the AV node fiber would get to threshold on its own. It would take a while, but it would get to threshold on its own. And once it got to threshold, what would happen? The action potential would travel down the AV node fibers into the ventricular fibers, and the right and left ventricle would still contract. The AV node is a backup. Okay? If the SA node were to stop working, then the AV node would begin getting to threshold on its own. The AV node would start generating its own action potentials, and those action potentials would then propagate into the ventricular fibers, and the right and left ventricle would still be able to contract. And this would, you might think to yourself, well, if the AV node, it takes the AV node a lot longer to get to threshold. Okay? Here's what we can say. We know this approximately, that the AV node generates around 40 action potentials per minute on its own. And the SA node is more like 70. Its intrinsic rate is actually 100. Talk about that later on. But that's what you get. Like if it takes the AV node longer to get to threshold, the number of action potentials the AV node will generate per minute is obviously going to be less than the SA node if the SA node gets to threshold faster. So the AV node can generate on its own about 40 action potentials per minute. Okay. So what type of heart rate is this going to translate into? If the SA node stops generating action potentials, then the action potentials won't rip throughout the atrial fibers, and the right and left atrium will no longer contract. But that will enable the AV node to get to threshold on its own about 40 times per minute. And that will cause ventricular contractions to occur about 40 times per minute. And then oftentimes in these circumstances, your sympathetic nervous system can kick in, as you're about to see, and actually pick up that rate a little bit. And point is, the person can actually be okay for a short period of time. Because how does most of the blood actually get from the right and left atrium into the right and left ventricle. It travels from the right and left atrium into the right and left ventricle actually just through, a pressure gradient. Like the contraction of the right and left atrium actually only pushes like the remaining 20% of blood or so into the right and left ventricle. So, you can survive for quite some time without your right and left atrium contracting at all. And the reason that you can survive is because, well, the AV node can take over. The AV node is kind of the secondary pacemaker of the heart, right? The SA node is the primary pacemaker. It sets the rate of the heart because it generates the most frequent action potentials per minute. But if it gives up its ghost, the AV node can temporarily, hopefully temporarily, take its place, and the ventricles can still continue to contract. So we can conclude that the AV node is actually important for two reasons. Probably more, but two big reasons in this lecture at least. The AV node, for one, accounts for the delay, right? The right and left atrium contract, then there's a break. Because the action potential propagates down the AV node fibers so slowly, and then moves into the ventricular fibers, and then the ventricular fibers contract. Okay, so the AV node accounts for the delay that occurs. And I don't even think I mentioned this. Like, what's the physiological significance of the delay? Why do you want a delay in the first place? And of course, the answer is you want the right and left atrium to contract before the left and right ventricles contract, because you want the blood to be finished being pushed from the right and left atrium into the right and left ventricle. Okay, obviously, I shouldn't say obviously, but that's the explanation. You need the right and left atrium to contract first, so that the remainder of the blood can actually get pushed from the right and left atrium into the right and left ventricle before the right and left ventricle contract. So you want there to be a delay. Okay, great. That's why you want there to be a delay, but this is the physiological explanation, we think, as to why the delay exists. So the AV node is important for causing this delay. What's the second thing that the AV node is also very important for? Well, if the SA node stops working, the AV node can take over. Yes, it's going to generate less action potentials per minute than the SA node will, but it's better than not having a heartbeat at all. Okay, so you'll see that in the heart, if you take more advanced physiology, especially cardiovascular physiology, there's lots of backstops that the heart has the evolutionary process is clear in that the heart evolved to account for many possible problems that can happen. And I mean, if you think about like a plane, planes have so many backup engines, and that's obviously for the intention of, if one of the engine goes out, and you don't want the plane to go down because of one, faulty engine. There's many within a plane. It's the same thing here. Like if the SA node were to stop working, does the person just die? Like the heart has has evolved to have has many backup mechanisms to deal with issues that come about throughout the course of a lifetime. So that's what I have to say about the AV node, and now we're going to talk about conduction fibers. So what are conduction fibers? Conduction fibers are depicted in yellow in this image. They look like it's like a neuronal system that exists throughout the heart, or it's some specialized type of like, tissue that exists within the heart. Everything in yellow here, these are supposed to be conduction fibers. Okay. Conduction fibers are just cardiac muscle fibers. Okay. Yet again, every textbook does this. They draw the conduction fibers in some crazy, mysterious way, and it creates mass confusion. Everything you're seeing in yellow, okay, whether it's actually the SA node, the AV node, or these conduction fibers that you're seeing, they're all cardiac muscle fibers. Okay, those are cardiac muscle fibers, but they serve a little bit of a different role. Okay, what conduction fibers do is, first of all, they're attached to regular contractile fibers, the conduction fibers. I'll go through this in a second. But the difference is that the conduction fibers can propagate their action potentials at enormously fast rates. Okay, so if you look here, right, here's the AV node. Where does the action potential go after the AV node? It travels into what we can say are let's just call them ventricular fibers. What it's actually traveling into are conduction fibers that are part of, they're part of the right and left ventricle. So we're going to have to draw this to make sense of it. So here's what we're going to do. These are cardiac fibers of the ventricles. This is within the interventricular septum. The ventricular muscle that exists right in the center, okay, kind of between where the right and left ventricle is. The interventricular muscle, we'll call it. Okay, here are some fibers. Okay. What is important to realize about cardiac muscle is it branches. What does that actually mean? It means that one cardiac fiber actually attaches to many other cardiac fibers, right? There's a branch, okay? And then this one is going to attach, this cardiac fiber is going to attach to a whole bunch of other cardiac fibers. This one here is going to attach to other cardiac fibers. This one here is going to attach to multiple other cardiac fibers, right? Cardiac muscle fibers branch. All right, we'll draw some more here too. I realize the last ones I drew, they didn't branch, but whatever. Okay. Let's say again that this is muscle of the interventricular kind of septum. The interventricular septum is actually made up of both cardiac muscle and some connective tissue too, but that's a whole different conversation as well. So anyways, what we're going to find in there are both conduction fibers and regular contractile fibers. We're going to say that these two, we can even add another one here. These three right here, let's highlight them in red. These three are conduction fibers. These ones, how about in green, are regular contractile fibers. All right. Conduction fibers. What conduction fibers do is, well, first of all, this conduction fiber ultimately has had an action potential passed to it by somewhere back in line the AV node. Okay, one of the AV node fibers, right, passed an action potential to probably another conduction fiber, and the action potential has been propagating throughout all of these cardiac fibers. Okay. Conduction fibers have less actin and myosin, and it would appear that their gating properties, like they probably have like really high concentrations of voltage-gated sodium channels. There's all kinds of things. Their diameter is larger. The point is, conduction fibers can propagate action potentials at really rapid rates, and they don't contract that much. There's not a ton of actin and myosin inside, and they have multiple properties about them that allow them to propagate action potentials at rapid speeds. So here's what will happen. If this conduction fiber is propagating an action potential, first of all, the action potential is going to propagate down the conduction fiber incredibly fast. And where's the action potential going to propagate into? Well, in this case, it's going to propagate into two contractile fibers and another conduction fiber, because it's going to move into this fiber, and this fiber. It's moving from the conduction fiber into two contractile fibers and then another conduction fiber. And then where does the action potential propagate down this conduction fiber, right? The action potential is going to propagate down this conduction fiber incredibly fast, and then it's going to propagate into two contractile fibers and then another conduction fiber. The contractile fibers are just the regular contractile fibers that serve the purpose of contracting, the kind that we've been really talking about this entire time. Now, what's the purpose of the action potential propagating throughout any of these conduction fibers in the first place? And we can go back to the previous image and make perfect sense of it, I think. All right, in yellow, these are all conduction fibers. And you have to realize all of these conduction fibers, right, this is a simplified image, all of these conduction fibers are all attached to other conduction fibers, but they're also attached to the regular contractile fibers, which are more so in red, throughout the image. So the conduction fibers are attached to other conduction fibers, and they're also attached to contractile fibers. So what's going to happen here? The action potential is going to rip throughout all of these conduction fibers at an enormously fast speed, because that's what conduction fibers do. They propagate action potentials incredibly fast. So the action potential is going to travel throughout all these conduction fibers incredibly quickly. And in doing so, what's the action potential also going to propagate into? The action potential is going to travel from the conduction fibers also into the contractile fibers, because the conduction fibers are attached to the contractile fibers too. What does this allow for? What's the purpose of the conduction fibers? The purpose of the conduction fibers is to ensure that the action potential rips throughout the entire left and right ventricles fast enough so that the entire right and left ventricle can contract at the same time. There's an enormous amount of muscle that exists in the left and right ventricle, meaning there's an enormous number of muscle fibers. And the objective here is to get the entire left and right ventricle to contract all of the fibers basically in unison. In order to be able to do that, you're going to need this action potential to rip throughout the left and right ventricle enormously fast and spread that action potential to all of these fibers simultaneously so that they all can pretty much contract in unison. And that's exactly the job of the conduction fibers. The conduction fibers ensure that the entire left and right ventricle contract at the same time. All the fibers. To drive this point home, imagine that there were no conduction fibers. Get rid of them. All that is within the left and right ventricle are contractile fibers. That's all that's there. It's all red, no yellow whatsoever. Okay? What would happen? Well, first of all of the contractile fibers are attached to each other through intercalated discs and thus gap junctions. So the action potential would still propagate throughout all of the contractile fibers. Right? It still would. But the contractile fibers propagate action potentials fast, but not nearly as fast as the conduction fibers do. So what would end up happening here is the interventricular septum muscle would contract first, and then the muscle of the apex would contract, and then the muscle of kind of the upper left and right ventricles would contract, right? Because what would happen is the action potential would still propagate throughout all of these fibers, but it would propagate throughout them much more slowly. Right? There's so many cardiac muscle fibers throughout the, throughout all of the ventricles. The action potential would still propagate throughout all of them. But since they propagate action potential so much more slowly than conduction fibers, what you would end up with is a non-synchronous contraction. Different parts of the ventricles would contract at different times, which would be terrible because that would result in less force production and less blood would get pumped out into the aorta and out into the pulmonary trunk. So really what you need for sufficient amounts of force to be generated is you need the entire left and right ventricle to contract in unison. And what enables that to happen are these conduction fibers. The conduction fibers don't really contract to any meaningful degree. They do a little bit, but it's insignificant. The main thing that they do is propagate these rapid action potentials, and that ensures that the action potentials spread in to all of the contractile fibers of the left and right ventricles at approximately the same time, and that enables for all of these fibers to contract in unison. All of the contractile fibers of the left and right ventricles to contract in unison. Now, some of this we're not going to get into, but different conduction fibers are named different things. Okay, so for example, you have the conduction fibers, kind of the beginning of the conduction fibers after the AV node. They're called the bundle of His. Then the bundle of His branches in the interventricular septum, and it's called the right and left bundle branches, or just the bundle branches sometimes. And then after you get beyond the apex, these conduction fibers are called Purkinje fibers. There are some differences between the bundle of His, bundle branches, and Purkinje fibers that you might discuss like in a really advanced cardiovascular physiology course, but not something that we're going to do. Functionally, they're incredibly similar. Okay, but that leads into the whole conversation about, well, what's the whole conduction system? I really dislike the term conduction system, and I just don't like to think in those terms. People, when they talk about the whole conduction system, what they do is they include the SA node, the AV node, the bundle of His, the bundle branches, and the Purkinje fibers. That's what you remember if you talk about the conduction system. But I'm not a fan. To consider the SA node and the AV node to really be part of the conduction system, or have any relationship to these conduction fibers, I'm just not sold on. I get that there's a relationship, but I mean, like, to lump them in as the same thing, it just doesn't make any sense to me. So this is the way I oftentimes think about it. The way that a whole heart is going to contract is like this. The SA node fibers are going to generate an action potential. That action potential is going to rip throughout all of the atrial contractile fibers. And now there are probably conduction fibers that exist throughout the right and left atrial muscle as well, as is depicted here, and they would ensure that the action potential travels throughout all of the atrial fibers fast enough so that they all can contract in unison. Okay, fine. And then what happens is the action potential travels from atrial fibers into the AV node fibers. So what you'll hear is that the action potential travels from the SA node to the AV node. And it's like, well, sure, kind of. What really happens is the action potential travels throughout all of the atrial fibers, and then the one place where the atrial fibers meet the ventricular fibers is at the AV node. So the action potential travels from atrial fibers into the AV node fibers, and then the speed of the action potential slows down significantly through the AV node fibers, and that ensures that the right and left ventricular muscle can finish contracting before the action potential then starts propagating into the ventricular fibers. Okay? And then what happens is after the action potential gets past the AV node fibers, it enters into ultimately the ventricular fibers. And it propagates throughout all of the ventricular fibers at an incredibly rapid rate thanks to these conduction fibers, the bundle of His, the bundle branches, the Purkinje fibers. Okay? That's really what they do. You memorize that order, but what the bundle of His, the bundle branches, the Purkinje fibers do is they ensure that the action potential rips throughout all of these ventricular cardiac fibers fast enough so that they all pretty much can contract in unison. And then that's what causes ultimately, of course, the left and right ventricle to contract after the left and right atrium contract. So that is the sequence of events that takes place with a heart contraction. So that leads to the question of, with regard to the quote-unquote conduction system, is there a benefit to understanding the path? And of course, the answer to that is yes. I just don't agree with some of the terminology. So if we think about it, the action potential I've said all of this, but the action potential starts in the SA node. It travels throughout all of the atrial fibers. Okay, that's what's being depicted here and here. And then what happens is the action potential approaches the AV node. And from the AV node, the action potential propagates first throughout the interventricular septum, down towards the apex, and then from the apex up into the left and right ventricle. This is the general path that the action potential takes, from the AV node into the interventricular septum, down towards the apex, and then up towards the left and right ventricle. And the action potential, thanks to the conduction fibers, rips throughout this pathway so fast that all of the fibers are able to pretty much contract in unison. But I'm talking about what's the benefit of knowing the specific pathway, the specific sequence that the action potential takes, and that applies to an ECG. If you hook a negative electrode up to somebody's right arm and you hook a positive electrode up to somebody's left leg, what have you done? You've orientated the negative and positive electrode in the same fashion or in the same direction that the heart
is orientated, right? The heart lies on a slight left angle. The apex is slightly facing towards the left leg. And you've got a negative electrode on the right arm, and you've got a positive electrode on the left leg. This is what's called lead 2 of an electrocardiogram or an ECG. Now, ECGs are another thing, incredibly complicated. But what you'll hear, which is kind of true, but there's so much more to discuss with regard to it, is that as an action potential moves towards the positive electrode on the left leg, you get upward deflections, which is kind of true. I mean, it's sort of true, but there's a whole physiology to understanding it that is way more complicated than that. If you want to remember that, that's fine. But what is actually happening is these two electrodes are measuring voltage differences between the two ends, and whether it's more positive or negative based upon the interstitial fluid, based upon the action potentials, is what dictates the upward and downward deflections. It's way more sophisticated than just saying that if the action potential moves towards the positive electrode, you get an upward deflection. But that's how it's presented to people in more introductory and intermediate courses. Okay, so that's what we'll do. Knowing that, approximately, if the action potential moves towards the positive electrode, you'll get an upward deflection. That enables us to understand an ECG. So this is an ECG, specifically lead 2. And what starts off is what has to happen. First of all, the SA node generates the action potential, then the action potential propagates throughout all of the atrial fibers. As the action potential propagates throughout all those atrial contractile fibers, it's technically headed mostly in the direction of the left leg, right, towards the positive electrode. So we get this upward deflection that is referred to as the P wave. Okay? Then there's a delay, and then we see what's called the QRS complex. The QRS complex is mostly an upward deflection, although you should notice there are some downward deflections in there, which is very complicated to explain unless you were to go into very detailed electrophysiology related to electrodes, and of course we're not doing that. So the QRS complex is this mostly enormous upward deflection. Okay. What is the QRS complex? Well, it's the action potential propagating throughout the interventricular septum down towards the apex and then up towards the upper parts of the left and right ventricles. Okay. It's the whole thing, the whole action potential is mostly headed towards the positive electrode. So what you'll hear is that this mostly causes a very large upward deflection. Okay, so the QRS complex, if we want to simplify this as much as possible, it is the action potential propagating throughout the ventricular fibers, and thus it's the left and right ventricle contracting. Okay, and where's the pause coming from between the P wave and the QRS complex? Well, the pause is thanks to the action potential slowing down as it moves through those AV node fibers. Okay, so if we see the P wave, that means the atria have contracted, right? The left and right atrium contracted. Then there's a pause, and then there's the QRS complex, which tells us that the left and right ventricle have contracted. Okay, and then there's this pause, and then what we see at the last part of an ECG is we see the T wave. The T wave is where the ventricular muscle fibers are repolarizing, right? Because what are all of the ventricular muscle fibers right now? After the action potential has propagated throughout all the ventricular fibers, they would all be approximately, like positive 10, positive 20 on the inside. And then they need to repolarize back to negative 90. That exact process we didn't talk about because it was too much. But when all of those fibers repolarize, you see the T wave. And you'll notice that the T wave is an upward deflection. And there's a very... I'd love to discuss it in more detail, but you would think it should be a downward deflection technically, but it's actually an upward deflection. And that's because repolarization actually happens in the complete reverse order than you would expect it to. And that actually translates into an upward deflection. And that is the entire ECG. Maybe one thing that you do notice is that where's repolarization of the atrial contractile fibers, and the belief is that it's buried somewhere in the QRS complex. I shouldn't say belief, it is. The repolarization of the atrial contractile fibers the waveform gets buried in the QRS complex. And that is your standard ECG. The next time you or somebody else that you know has an ECG, hopefully you can look at it and you have an idea of what you're actually seeing. Right? You're seeing the action potential travel throughout different portions of your heart, which you can relate, and you can say, well, that means my atria are contracting, or my ventricles are contracting, or they're repolarizing, etc. And if you're interested in this on a much more detailed level, which is what it actually takes to fully explain all these upward and downward deflections, like I have said 20 times, take some more advanced courses. And that concludes this lecture. In the next lecture, we will start talking about some clinical values like stroke volume and cardiac output. Basically, you know, how much blood does the heart pump out and thus around the body, and then how the sympathetic and parasympathetic nervous system and other factors can influence it.